Speaker is going to turn a little bit the delivery route from oral to nasal. At the end of the day, they're all linked, somewhere in the middle. So, obviously, brain delivery and so on, another area, and nasal delivery is another area that are important in the drug delivery. Debanjan goes by name Deb, is a drug delivery scientist and an innovation-driven technology manager with over 19 years of product development experience in driving projects from concept to commercialization. He has marketed over 40 drugs. That's like more than two drugs per year. So that is great experience to share with us. He has been working on drug delivery strategies, scale-up expertise, and intranasal routes for drug delivery specifically, and bioadhesives, and there's a whole list of all the technologies that he has been involved. So, certainly wealth of information that we would benefit from. Currently, he serves as Senior Associate Director and Science Fellow at Bayer Health Care Global R&D, where he manages a team of scientists to bring innovative dosage forms by managing over 1 million plus formulation development budget. With that, I will ask Deb to come on stage and talk about intranasal delivery and how to enhance bioavailability there as well. Welcome. Thank you.
Thank you, Firoz. Thank you. So how is everybody doing? Can you hear me all right? Perfect. Yeah, as Firoz said, I'm going to take you to a different journey. I mean, we have been talking about processing and other things, but it has got some similarities as well with our last presentation by Ed. So let's get started. So essentially, what we are trying to do here is that there are some unmet consumer needs or unmet patient needs, and we are trying to address them. And right now, my focus is developing the nasal portfolio for Bayer. Okay. So it's kind of frozen. Yeah, all right. So nasal drug village, just a quick recap. So it has got some very specific advantages, like, it's got very fast absorption. And if you think about it, for some drugs, actually for most drugs, the nasal bioavailability is almost equal to the IV dosing with a little bit of time lag. In other words, if you have an IV dose established, you can transfer that to a nasal delivery. Now, of course, there are a lot of challenges as well. Now I'll go through them. But nasal is a very developing field, and I feel greatly privileged to to understand more of it. Okay, so complexities. I mean, nose is not the nose that you see from outside. Okay, it's a lot of stuff, a lot of anatomical nuances are inside. If you see the model yeah, that's my cursor. Yeah, so we use a silicone mold, but also we are doing some studies. I'll come back to it later. But just to give you a brief flavor of how much variable this organ is, if we just look at the two circles which I've made, the valve, the front portion, the turbinates, which are the middle portion, we have 3 turbinates, and the nasopharynx, which is the back side. From person to person, it varies about 39% and 29% respectively, which is more than like 70%. So you are targeting an organ which from person to person may vary as 70%. Nose is a very dynamic organ. Sometimes it's swollen, sometimes it's constricted. There's a bunch of things happening. So that makes it a really challenging organ to deliver to model your bioavailability. Okay, so some of the products we have, just wanted to show you and get it out of the way. These are some of the non-drug products. These are some of the drug products. We are pretty much available throughout the world. And now two products which we are focusing on right now for line extension, but making a platform technology. And what is unique about these two dosage forms is that you can get this over the counter, but there is a base technology which is very unique. And what we call that is a no-drip platform, meaning when you apply a dose inside the dose, kind of rhymes, it doesn't go off or run away. And what I mean by that is, if you look at the structure of the nose, once you apply a dose, part of it would be here in the turbinates, most of it. But if you apply too much, it will go back to your tongue region in the back, or it will simply come out in the front. So we have studies to show that we have lost as much as, I would say, 40% of the delivered dose. It just either went all the way in the back to your, it will go to your actually GI tract and will pass away. So that's how your anatomy is designed. And once it passes through this place, some of the drugs over here are so bitter that once you taste it, you're not going to use it again. So that's a real problem from the consumer perspective, so that's something we're addressing. But even more than that issue is you are losing drugs, you're losing bioavailability, you're losing dose altogether, so it's a big problem. Okay. So how do we modify this in the formulations? And one of the major challenges with nasal formulation is, and I'll come to that later on, if you think of 1 ml is too little, we have to deliver at least 0.2 ml. That's all you're allowed, maximum, in each nostril. That's like the maximum you can give, 0.2 mils. Ideally, you should give 0.1 mil or 100 microliters. So you're restricted severely by volume. All right? Now, on top of it, you have to make it modified release by adding a variety of things which will kind of enhance the formulation. So typically we use viscosity modifiers and surface active agents to modify the permeation and solubility. And then nasal ciliary movement is another important phenomena. Whenever there is something inside your nose, let's say dust particle or whatever, you have nasal cilia, especially in the middle part of it, in the ciliary movement will catch those foreign particles either by air or by liquid, whichever way it enters, and then it will propel it back to your GI tract to clear it off, and this clearance happens every 15 minutes. In other words, whatever you give inside your nose, an extraneous agent, is cleared off in 15 minutes. So you have only 15 minutes to influence the bioavailability absorption in a very small window, and therefore there are certain specialized formulations which can actually beat that, I would say the natural physiological way, that's the way we evolved, but kind of beat that to kind of increase the residence time in the nose. So we call them ciliostatic agents, viscosity builders somewhat act towards it. I'll go into more details on that. All right. So one of the easiest ways to kind of fix it is using a thixotropic polymer, which, if you buy one of these OTC products, you will see in the label, we have microcrystalline cellulose and sodium CMC. Unique properties at a certain percentage, very high viscosity, it's not shown here, it's around 80 CPS. It will drop to 10 CPS and go up to about 70 CPS within a matter of seconds with the application of stress. If it's in rest, it's a gel, shake it, flows, and stay it for like 3 seconds, 5 seconds, it's solid, back solid again. So what it does is, once you spray, the shear force in the spray device applies the shear force, and it is actuated as a liquid form, gel to sol, lands in your nose, goes back to gel. You can use the simple cellulosic components to make your formulation stick to the nose. I'm looking forward to this phenomena to use it as a, it increase of clearance rate. But the other side is that you don't have to actually, this formulation is not going to flow back in the side, and you will not feel the bitterness of the drug. So it will stay inside the nose, not like run away. Okay, so there are many other polymers we can use. Unfortunately, I wanted to show this because this is a sad reality. In IID, inactive ingredients database, we have only 7 polymers, those are listed. That's it. What you can see in the right-hand side, any polymer that you use for specialized purposes, you have to do elaborate tox studies. Even something like a polymer, hydroxypropyl cellulose, which is used for eye, ophthalmic uses, you can't even use it in the nose. So in FDA's eyes, your nose are more sensitive than the eyes. But whatever it is, this is the sad fact, and we are really constrained within this to make some new innovations come to light. Now, another part of the viscosity is bioadhesiveness, and what it does is it actually has a propensity towards the mucus layer, and there's a lot of studies about how certain materials stick to a mucus layer with there's some interactions, ionic, other things. This interfacial tension that comes into play. But overall, if you look at this, is the nose has about 15 mils of volume, but surface area, due to this microvilli, makes it 600-fold. So you have ample surface area, like about 96,000 centimeters square, to make your drug get absorbed. So it's a big surface area for a small organ. Okay, now going to this advanced delivery forms, what are the things that you can actually add? You can have mucoadhesives, as I just said, absorption enhancers, there's a bunch of them. Preservatives, of course, high viscosity gels, and then you go to the realm of micro, nanoemulsions, nanoparticles, liposomes, and even powders. So there are a bunch of things if you go to the literature, you will see that there are a lot of things people have tried. Some of them has worked really well. But our goal is to take things to the markets. I mean, something that is robust to manufacture, something that is repeatable, high quality and all those sort of things within a reasonable cost. So again, coming back to what we call as a matrix of it, there's different ways these polymers behave inside the nose. Some block polymers, which will gel in like body temperature, 37 degrees poloxamer is famous for them. It will form micelles and then it will turn into gels. There are other things like polysaccharide chains, like pectin, what you make jams and jellies with. If I throw some calcium ions, those are in the nasal fluid, it's going to gel, ion sensitive. Then chitosan, our famous bioadhesive stuff, derived from the shells of shellfish, yeah. But it's not soluble as it is, you have to acidify it, but nonetheless, it's a fantastic mucoadhesive agent, it just sticks like glue. Then we have gel and gums, carbopols, HPMC, host of other things. So you have to study these polymers, find optimized ratio, and you can use this so that these polymeric systems forms a matrix inside the nose once delivered and resist nasal clearance and delivers the drug over a period of time. Now, okay, I think this is a very old saying in the industry, from brick dust to blockbuster. We know it for oral, but what about nasal? Well, we're trying. So one of the things, we all know that amorphous solid dispersions is one way, lipid-based formulations is another way, and we just talked about it in the last presentation. So those are a couple of feasible ways which you can do that. The challenge is this. You only have 0.2 mils to deliver the dose, not even a full mil, 1/5 of a mil in each nose. That's the biggest challenge. So you have to develop a system which is extremely, extremely efficient in solubilization. Now, out of all this, we try to, there are particulates, liposomes, other things. As we settled on self-nanoemulsifying system, just a basically set system within the nano size, because it can be scaled up easily using a simple mixing process. And we kind of said, okay, this is kind of easy enough to manufacture. We can have good control over it. And so we kind of zeroed on that. And we took a few model drugs like BCS class 2, 3, and 4. And we tried to generate a model, like a base chassis model, where we can just throw in the drugs it will improve solubility and permeability. It's like, it's something which you don't have to reinvent the wheel every time. You might do minor modifications, but you will achieve your solubility and permeability targets. And the number of restrictions in that small volume that you have to have buffers, preservative, antioxidants, osmolarity, stability if you need that. And you only once per nostril 0.2, even this is pushing it. Usually it's 100 microliter per nostril per dose, so totally 200 microliter for both nostrils. To me, this was the most challenging thing. So you have to come with a system which is extremely powerful in solubilization. So you need very little water. Your water fraction has to be very, very small, so you have to accommodate the SNED system inside it. We tried both SNEDs, incorporated SNEDs into gels and sprays, but here the focus would be in sprays. It's very kind of straightforward to make it. We can do dissolution, we can do all sorts of in vitro studies. Advantages, obviously, solubility and bioavailability enhancement. We can go to any matrix, sprays or gels. You can stabilize sensitive drugs. Limitations, as I said, extremely small nasal volume. Sometimes there's also, if you throw a bunch of surfactants in your nose, it might be irritating. So that's a very critical thing to note. And there's another thing, difficulty to atomize. If you're using a spray device to atomize, then 50 CPS is kind of like cut off, but still 50 CPS, you have to go to a specialized manufacturer to get it. So 50 CPS is not much. If it's only 50 times, if water is 1 CPS, it's 50 times. It's not like honey flowing. It's most likely a thick polymerase solution. It's not super thick. And other part is that in order to spray, you can go up to, with all your excipients, up to a 50% level max. The rest 50% has to be water, otherwise you cannot spray it. It'll become gel, and if it becomes a gel, you have to do some additional studies. You can put it in here, which is external layers, then it has to diffuse inside. So spraying for me is the best option, but if you have a very thick material, thick liquid to spray, sometimes spray won't work. It will just jet out, or it will not come out at all. It's unsprayable. Okay, so what we did was we chose a bunch of surfactants based on HLB values, and if you see here, I have categorically omitted the oil, which is, and making it a type 4 type of SNEDs. If I add an oil, things get much easier to do. And why I did that is that if there's oil, unlike oral, nasal has limited enzyme systems. So the oil has to be digested in order to release the drug. So I didn't have the luxury. If you have an oil, it's really good because you can get very nice particle sizes. If 100 nanometers is no problem. But if you use a system like this, they're only held by steric interactions or ionic interactions. So they're a little bit bigger, about like 200 to 250 nanometers, but it doesn't need any digestion. It's more instantaneous. And we chose BCS classes of drugs with different solubilities and different log Ps, class 2, 3, and 4, just to see how robust is the system, the SNED system that we created. And these are the 5 surfactants that we used. Pegylated glycerides, twin 80, span 80, polyglycerol threolate, and citosterol glucosides. Now, okay, solubility is no problem. Throw in HPLC, measure it, good. So how do I go to in vitro permeability studies, correlating solubility with permeability in vitro? So one of the ways is there's a paper by Hendricks. So if you know of the PAMPA system, parallel artificial plate, it's used a lot in early screening process to kind of determine solubility to screen out drugs. And they use hundreds and hundreds of PAMPA plates like this to screen. They will have the drug in a DMS or water, and they will just push it through how much is coming from your acceptor to donor side. Very simple procedure, but we modified this PAMPA system based on a paper, and we modified it further. It's a PAMPA system to reflect permeability of drugs in a nasal world. So we did some modifications, and this paper is, you can find it anywhere. So we added mucin, we added phosphoryl choline, kind of changing the membrane that we have, membrane characteristics. and we used it for our screening purposes. So our focus was, to be honest, more on the permeability, because we know solubility, 20–30% increase is not a problem. We'll get it one way or the other. I'm more focused or more interested in how much permeability it will increase. Okay, so this is one of the ways we tested it. And secondly, we also did by France diffusion cells. I think everyone knows that. I didn't put a picture up there. So the first results are on Pampa, and the results below are on using France cells using porcine mucosa. And what we have seen is in Pampa, the increases were at let's say close to 4% or two times or 4 times increase. But using the France diffusion cells with actual mucosa, it was like 30% to one was a little low, about 6.41 and 46% increase in the flux. I haven't read actual permeability values, but we are more interested in the flux, because how fast can you get the action? So flux increased by 46% in a class 4 drug, previous class 4 drugs. This was very surprising to see this much of increase. And the profile looks something like this. The first one is, the one with the graphs in the 3, they are the SNEDS formulations, and similar ones is without the sense in phosphate buffer 6. So if you see the first circle, it's almost like instantaneous release. Then after a period of time, we ran it for 24 hours, but for instant relief, we usually go for only for 4 hours just to see. But I ran it all the way just to see what happens, how much drug is released, and what else you can find out. So after some time, like 240 minutes, it kind of becomes a plateau, and it releases the drug. And still the whole drug is not, I didn't show you the percentage release. Still there was about 15% drug left from the formulations. And how was it applied? It was applied as a nasal spray, which has SNEDS inside. So SNEDS incorporated nasal spray. Just take the dose, take the skin, spray it onto it. And then you measure how much is coming out over time. Okay, now I'm going to shift a little bit towards a different angle. So far we have talked about drugs in nasal. So where does it go? If you put a drug in your nose, it will act in 4 different ways. Nose to nose, which is local, okay? Your oxymetazoline sprays. Nose to blood for systemic, all right? Nose to brain is by a pathway by olfactory nerves and trigeminal nerves and lymphatic pathways, nose straight to brain. And then nose to blood to brain. So if you know about Narcan and Spravato, the ketamine sprays, they are nose to blood to brain, not nose to brain directly. Some of it goes nose to brain, but predominantly drugs that are in market now for nose to brain, they can cross blood-brain barrier by itself. So it's nose to blood to brain, crossing the blood-brain barrier. You can do SNEDS with this. And just comparing with if you are specifically looking to target this olfactory bulb region, usually, just to give you a comparison, in oral, the drug surfactant blend and water ratio, you can go about 1:30:30. Here, you need to have 1:5:5. Or basically, you need almost like a super SNEDS, which will have 6 times more solubility, oral versus nasal. So when we did that, we think about, okay, what we made was some sort of a micelle. It's not, it's a little bigger than a micelle, I would say. It's not a nanomicelle, but can we do a nanomicelle? That was our thought. And especially, what would be the in vitro models to test it, because there isn't any. If you want to study even nose to blood, there is no in vitro model. But nose to brain, even harder. And this is, again, kind of telling you how this is the olfactory bulb that you are seeing in the top, this nerve, in the top of your nose, in the upper turbinates. You target there. The olfactory bulb is its only neuronal structure which is exposed to the outer atmosphere. It's in direct contact with the atmosphere. That's why you can smell. So you target that, and it will take your formulation, with the formulation drug inside directly to your brain region. And it happens by 3 pathways, but I focused on olfactory because that's more a defined target, but there are other routes of targeting as well. Okay, so I was talking about olfactory in my last slide, this one. So you will see the top of the nose. So we had a model like this, what we call as a cocaine model made from Japan. It is a silicone-based model where you target the formulation and spray it and see where it's landing up. The picture all the way in the bottom is something which doesn't have the modifying polymers or viscosity builders of those. It's spread all over the nose. You will see even some part of it is flowing out, flowing back. However, if you can angle the nose at about 60 degrees or even 75 degrees, your deposition will be in the top part of the nose and where the olfactory bulbs are located, and you can enhance your chances of nose to directly to brain delivery. And this is just a model we try to do with angle of actuation and deposition in nasal terminates. You can actually even model it. Let's see. Okay, so let me show you some nice videos now. All right, this is not firecracker in Diwali. So this is, let me see. Okay, this is what we call as plume, which is if you see at a spray, if you see from one side, see how it looks like. This one is a little bit thicker with polymeric. It goes out like with a thinner angle. This one is more diffuse. It covers a larger area. This is even thinner. This is a function of the viscosity and the mucoadhesive polymers. The more you add, the more thicker the substance gets. It's very difficult to spray out. You can have custom-made sprays, but they're very expensive. If you use a normal commercial available spray by one of our regular manufacturers, that's what's going to happen. So similarly, again, control. What this is called as spray pattern, where you look at the spray from the top, and you can see that circle again. So this is basically you capture these images by laser from the top. And if you see the top profile of this one, a lot smaller, even smaller. So what if the bigger ones are about 39, 40 microns, and the larger ones are huge. They're like 541 microns. Now, what does it actually do? What does it actually mean? So it means that if you want to fit everything into a model, these parameters actually influence in which part of the nose your dosage is getting deposited. And if you're interested to deposit your formulation in a particular area of the nose, you can follow this and make it land. For example, if you're targeting only the upper part of the nose, all right, here's the way to do it. All the whole nose widespread, here's the way to do it. So we kind of see how the SNAD's percentage will work out, how the mucoadhesive part is, how the concentration of that one influences it, what is the actuation angle, and then the viscosity, density, the plume angles, what you saw, droplet sizes, and then we chose the turbinates. Superior turbinate is the portion up, then middle, and then inferior. So you can model by studying different characteristics. So it's not just the formulation of viscosity and mucoadhesive agents. It's also the spray pump characteristics, which are also important. So it's a drug-device combination. These are our latest fancy things that we're trying to do. So we are real-time, we're trying to see when we spray it, and if I see my whole skull, this is a CFD, computational fluid dynamics. We are still in the early phases, but this is what the concept is, to see real time how the deposition is going to take place. So for example, all right, so you had a spray. This is how the particles behave, and you can see still this is normal, we're just showing is just water. That's why it's not sticking inside, but if you take water as a standard, this is how it's going to behave. Fine plume, bigger plume goes inside, but then at least 40% of it is down your throat. So this helps us to kind of real-time actually which part, which section, which gets the maximum deposition and all those things. Sorry. Okay, so then I'm actually in the close. So as I said, there are 4 types of modified nasal delivery system. One is nose to nose, which is local, nose to blood, which is systemic, and nose to brain, which is direct, and then nose to blood, and then to brain. For the last two, we are still kind of work on process, but for the nose locally, there's nose to nose and nose to blood, we have fairly good understanding. The field has enhanced quite a lot. We have local targets, systemic targets, some vaccines we're delivering by nose, PKPD models widely available, you know the kinetics, and devices available. So you can see me sleeping in one side. The other side, there is a unmet need of addressing a lot of brain disorders that are coming to the market, and we are thinking the next 20 years we'll be probably using formulation devices or formulation ways how to address brain disorders. And that's why nose to brain takes kind of precedence. Problem is, each drug, unless its solubility and permeability is increased to a very high level, and it can be targeted to those nerve punches for direct uptake, it is really difficult. There are no in vitro models available. We have to develop computational fluid dynamic studies and other things. I'm also developing a neural model where I can gauge not just permeation through the mucosa, but straight, if I hit it in the brain, if I hit a nerve bunch, like a bunch of neurons, how will SNEDs affect the uptake of the drug directly to the neurons? So SNEDs neuron interaction. It's tough working on it, so you can still see me kind of not getting my full night's sleep, but we should get there. So I think I'm coming to the end. This is my LinkedIn profile. I didn't get my card, so nowadays everybody's going digital. Feel free to get in touch. And this is my team, and hope you enjoyed the presentation. Any questions?
Any questions for Deb? Yes, Irfan.
Hi, very interesting presentation. So I'm coming from a solid oral perspective, so I just want to understand, you said there's like about 7 polymers available. The limitations in polymers for nasal delivery, is it because part of it goes to the lungs? And if the polymer is not biodegradable, then there might be challenges, or is there more challenges that stops use of other polymers?
Yeah, a very good question. So I think there are people here who are more aware. First of all, this is with the FDA's IID list, nothing with BAL. There are polymers much better performing, which I can use in the lab. But if I have to go rapidly to market, FDA's IID list kind of lists 7 of them. I think, and correct me if I'm wrong, just asking you openly, this list is generated by FDA based on the number of NDAs that get submitted. They just pick up the ones that's for nose, and they just publish it. Even for that, I think you need permission from the sponsor. So that's how this IID list is generated. I mean, maybe they've been lazy, or somebody didn't want to use special polymers and apply in the NDA. I don't know, but that's what we got. But to answer your question, there are polymers way superior than this 7 of them.
Yes, Sebastian.
Hi, I'm Sebastian from AbbVie. Thanks a lot for the talk. I'm not from the area, I don't have experience in that area. I was just wondering if these very nice CFD methods you've mentioned to better understand delivery and deposition, can that kind of work benefit from pulmonary delivery to the lung? You know, studies that happened maybe 2 or 3 decades or 4 decades ago on understanding how products and not just powders, but also liquid formulations can be delivered to the lung.
Yeah, I think to answer your question, the people who made the CFD actually he was a naval engineer, so he doesn't even know what the interior of the nose needs. What it only needs is some physical characteristics and internal structure, a diagram which either generate from a cast, and then it can be transferred digitally by vector images. For lungs, the only issue I would think is that lungs is dynamic, going up and down, but if you can make an average of lung space, how it look like the structure, and you have to make an exact 3D coordinates of it, then it's highly possible. but I'm not a CFD expert, I have to admit. I've just seen people use it, and I know how it works, but there's an elaborate code that you have to write for that.
All right, any final questions for Deb before we move on? No? All right, thanks very much, Deb.
Thank you.